back. I had finally finished school. Yeah, I've been doing this whole thing while doing finals and stuff. So it's been a little bit because I was doing finals. But we're done. I have about 20 days till my test. So the goal is this week I get all the biochem done. Next week I get all the biology done. And then gen chem and then physics. I should be able to fit all of it. So I would if and then ideally I get to do what's the name? Psych Soc. So but we'll see. So chapter 1.4 and 1.5 is all about protein structure. So section 1.4 focuses a lot on primary and secondary structure. So the linear sequence of amino acids is primary structure. And that means like if we had something like GOU, ASP, I don't know, MET, and so on and so on. This is your primary structure, okay? So again, stabilized by peptide bonds, and we know that a peptide bond would be something like this, okay? And then so on and so on, okay? And the reason why this stabilizes it is due to the freedom of rotation. So this has sp2 character. So it can move around this. So that's what's making it stable. And yes, this determines all high level protein structures. Okay, and we always read it N to C. Okay, meaning that we go from like N to C. Okay, you always go like N to C. So your N terminus is always at the end. So your M terminus should always be at the left side and your C should be at the right side. So secondary structure is pretty much this protein structure, but now we have multiple. So it's like we have this also connected to this or something like that. There's a few types. Alpha helix is one of there's two main common ones that we only care about. We care about alpha helix and beta sheet. And again, you could only imagine a helical. This is not an alpha helix, okay, but but you could only imagine, right? It's going to be a right hand coil between different peptides. Okay, so it's the stacking of multiple of those in the shape of a coil. You know that your R groups stick out, and it's found in keratin, which is this is in your hair, and then different transmembrane proteins. So beta sheets are chains lying next to each other. They could be parallel or anti-parallel. So that means that parallel or anti. They could be found in two different forms. It's pretty much the significance of that. They're found in silk, and then the R groups alternate above and below. So one thing is we need to talk about proline, big star, very important. So proline induces kinks, meaning that it sort of messes up the secondary structure. So you'll see it in turns, but you're not going to see it in helices or sheets. Okay, so this is definitely a double start. Yeah, so definitely understand this. This is your basic level of protein structure. So now let's finish up our protein structure by going into secondary and tertiary structure. So 3D shape of a single polypeptide, stabilized by interactions among side chains. And these aren't just hydrogen bond interactions, right? We're talking about salt bridges, ionic interactions, all of these. And among different side chains, meaning that like different types of amino acids. So like if you have glutamine reacting with that or cysteine reacting with itself, right? So if you have two cysteine molecules, those two will always form a salt bridge, okay? And that's the same thing, interactions among side chains, okay? So next we have, and then part of, again, more interactions, right? You could have hydrophobic interactions, right? Non-polar group goes inwards. We call this the hydrophobic collapse. Um, Definitely, again, this is huge. Hydrophobic collapse is pretty important. So your non-polar proteins are going to go on the inside, and all your polar ones are going to go on the outside. Okay, and like I said before, we have salt bridges, hydrogen bonds, and remember, every molecule has these van Waals. And then we also have that exactly. That's what I talk about here, cysteine residues. Okay. And yes, major driver, nonpolar. Again, there's a reason why I said this twice. So we need to talk about denaturing. So usually it's caused by heat, and when you denature a protein, you unfold it. So if you heat up a protein, it's going to unfold, and it's going to go. You're going to lose your tertiary structure and revert back to one of these two. And last but not least, we have quaternary structure. So not all proteins have quaternary structure. Multiple polypeptide subunit into one functional unit. Same forces as tertiary structure. Again, it's just more. So it's like two tertiary structures together. So one example are your antibodies. These are a type of quaternary structure. They're produced by B cells. They're part of the adapted immune system. They bind to antigens. And again, this is heavy understanding. Structure of antibodies, we should all know there's two light chains, two heavy chains. And again, you have multiple different units. So I only bring up that example because one, antibodies are very important, and two, it is a form of quaternary structure. So next chapter, chapter 2.1, we're going to get into enzymes. So I will see you there.